Hi everybody, I am Jitendra Kumar Biswal and I am working as a scientist at ICR, Director of FMD. Those who are not knowing about the profile of our institute is mainly we are dealing with the epidemiology, more molecular epidemiology, serology, diagnosis and selection of the new vaccine strain of the foot and mouth disease virus. So we are the only institutes in India, which in throughout the world also, which exclusively dealing with FMD only. Sorry, is that the next one? Okay. Yeah. So we took a collaborative project between USDA, USA, and ICAR, DFMD to study about the virus persistence in natural conditions in different ecosystems, starting from the at the height of 7,000 feet from the sea level to the nearest to the in different tropical and subtropical conditions and different temperature conditions also. The main thing to answer is that what is the role of the carrier virus, carrier animals to spread FMD to naive animals? Is there any role? Because until now there are not enough data is available with respect to the suspicious role of the persistently infected animals to transmit the infections. And second thing is that to quantify the risk factors, how much risk they possess and what is the duration of the carrier status. And finally, we are also interested to find out some of the basic questions and some of the development in the basic biology of the FMD virus. So keeping these mandates in view, we have selected different places. But in this presentation, I'm going to present at one site in organized dairy, which is it's located in the central part of India, and it is the largest dairy in the central part of India, with a status of about 5,000 animals, both cattle and buffalo, swamp buffalo, domesticated buffalo. And during the time of the outbreak happened in the early part, the Jan of 2014, and at that time, approximately 1836 animals are on the milch, and out of 1836 animals, 222 animals has been infected during the course of the 5 weeks, which I can show here. So, sorry. Oh. Sorry about that. Yeah, it's okay. So, out of the 222 animals, means out of 1856 animals, 222 animals has been infected within the prevalence of 12%, which is quite acceptable in case of biannual vaccination period, because as you all know, that vaccine is not the magic bullet to prevent all the animals from infections. And during the study period, we have collected about 21 clinical affected and 16 asymptomatic cattle, and 11 clinical affected and 6 asymptomatic buffalo period. And we collect the samples at 2 to 3 months interval. The major challenge is to collect the OP fluids with respect to logistic, because depending on the discretion of the farm manager, those animals are not available each and every time to collect the samples, because sometimes they sell the animals also, and sometimes the animals also not cooperative to collect the samples, because it is hugely, incredibly difficult to collect the samples in the field condition, especially in buffalo. And after taking the OP fluids, we have transport to the central laboratory, which is about, take an example of 1,500 kilometers from the study site, and to liquid nitrogen, and we revive the virus in LLBK cells having alpha-1 integrin cell line, because these cell lines is considered to be equally as sensitive as the BTY cell. And at passage level 3 and 4, we are able to sequence the P1, the capsid region of the virus. So if you see the genome detections, I have divided the presence of the genome in cattle and buffalo. Until 13 months, we are going to find the genome. Of course, the first 3 to 5 months, the proportion of the animals found positive with respect to genome detection by real-time PCR is more, both in cattle and buffalo, and it is drastically reduced down during the course of period. And after 13 months, in the 14 months to 2 years, means 24 months, these animals become entirely negative. With respect to the virus isolate, the story is entirely different. The virus isolate could be possible until up to 7 months in case of cattle. While in case of buffalo, we are able to isolate the virus for 13 months. So that indicates that buffalo can be a good incubator to harbor the carrier viruses as compared to cattle. Of course, the data are limited with respect to one farm only, but from the data, it seems to be that the extent of getting the virus from the buffalo is more as compared to the cattle. But if you find a detection of the genome and the virus isolation, there is a contrast, because in genome detection, we are able to find the genome positive until 13 months, but in case of virus isolation, they are found positive until 7 months. So, which substances also corroborate the earlier finding in case of laboratory animals also, because there are many publications which showing that though the animals are found in genome positive, but that doesn't mean that it is always possible to get the live virus from those carrier animals. It is the same as the virus isolation study. So, and when you sequence the P1 sequence of the carrier with respect to the acute virus, all of them are grouped into the in 2001 group, and more specifically in 2001D. I would like to inform you that in 2001 has been there is uprise in 2001, and then it get eclipsed by Pan-Asia until 2007 and 8, and again in 2007-8 there is a upsurge of in 2001 viruses, and now it has been completely dominated by in 2001 only. We don't have Pan-Asia in India now, and all the acute and carrier viruses are grouped in together. And if you find the nucleotide divergence from the carrier viruses isolated from the cattle and buffalo with respect to the acute viruses, the difference is with respect to the synonymous and non-synonymous to synonymous the changes. The highest changes from one animal is 0.357, and the lowest is 0.691. And with respect to the changes from the acute virus and the carrier virus, the highest percentage is 1.3%, the lowest is 0.01. So what it actually says is that it varies from animal to animals. That means the pressure or the extent of replications or the other factors, it varies from the host rather than the virus, because the data varies from animal to animal. And the same thing here, that the synonymous mutation is more as compared to the non-synonymous mutations, which seems to that there is a strong purifying selections due to evolutionary constraint. With respect to the nucleotide polymorphism, we found the maximum nucleotide polymorphism, the single nucleotide polymorphism, and followed by a polymorphism nucleotide at two sides, at the second site. And with respect to the amino acids is concerned, we found at 75 positions there are changes, and 66 positions are alternatively 1 or 2, and at 9 positions we have more than 2 amino acid positions. And the majority of changes in the BP1 followed by BP3 and BP2. With respect to the selection pressure analysis, with respect to by using different bioinformatics software, we constantly found that the 2 amino acids, BP1 138, 148, and BP2 78 and BP3 76, and positive selections. And we also found the 28 codon having episodic selections, including in BP4 regions. Also, we found BP4 61 that is episodic selection. With respect to the statistical parsimonies analysis is concerned, from all the viruses which we got from the carrier animals, 6 viruses, they have a nucleotide divergence more than 1%. So what it actually states is that whether in these 6 animals there is a high rate of mutations, or there is an introduction of the new viruses which you don't know during silent introductions, or during the course of the infections, during the initial infection, maybe the virus infected is a mixture of the different viruses, and they are replicating in independent rate. So that these 6 animals having a nucleotide divergence more than 1%, while the rest of the viruses are grouped together. And the one more thing, if you go to the extreme right-hand side, you can see that the last viruses which has been collected, from the March and from the August, they are connected to each other. So we have reason to believe that maybe from that buffalo, that symptomatic buffalo, it has been transferred to the asymptomatic animals. With respect to antigenic analysis, we found that during the course of the period, from the buffalo, there is a huge change in antigenic analysis. Initially in the March period, they all are homologous in nature, having R value more than 0.3. While at the end of the period, if you can see that graph, then they are antigenically divergent from the acute viruses. So from this analysis, we found that in case of buffalo, the antigenic divergence is more as compared to the cattle. So the final summary is that the mutation rate that varies from animal to animal. So we believe there is the host pressure is more than the viral pressure, and the mutation level is higher as compared to the normal acute viruses. And as you can see that the mutation rate is 1.816 substitution per site per year, which is normally more than that in case of field isolates. With respect to the antigenic analysis, value indicators that there is a fluctuation of the antigenic relationships between the course of the time period, and there is evidence of viral activity in persistently infected animals. So we believe that there is a host vector responsible for shaping the evolution of the carrier viruses. So finally, I would like to thank the owner of the CEO of the Addis Dairy and from ICAR, and our collaborator USDA, especially Dr. Louis and Dr. Jonathan, and funding from the USDA, and all the scientific staffs of ICRPD FMD to make this work happen. And finally, also I would like to state that me and my colleagues are here for the 3 days. So if you have any questions and queries, not with respect to this one, but entire the FMD situation in India, you are most welcome. And that's all. Thank you.
Thank you, Jitendra. We really appreciate it. And I see you had structures there, so if anyone had questions about where those mutations are located, then you, please come and talk to Jitendra. So thank you very much. Our last speaker, I see